Good afternoon. I'm going to steal the first line from something I just overheard. So I'm going to steal this brief line. She said, "Thank you for being here on a Friday afternoon when you could be doing something else. We appreciate your presence here." And before I get into the other business of the day, I want to first start off by thanking everyone who helped organize today's event. So thank you to Denise of McKinnon, who is our administrative assistant, Shavana's Porter, who is in the biology department, and the IT staff that has taken the time to do this for us. So thanks to them, and thanks to you all for being here. So let me get to the other part of the first 5 minutes. Who am I? I'm Dr. Aditi Pai. I'm in the biology department. Most of you know me, but I said who am I deliberately because I wanted to provide some context for why we are all here today. Most of you know this, but some of you don't. This is around the Who Am I project, or the Genes and Genealogy project, which is designed for undergraduate biology majors. How many of you are biology majors that are in the room? So it's exactly designed for you. And for that reason, I wanted to tell the others in the room what this is about. This project starts by inviting first-year freshman biology majors to ask the question, who am I? And we are asking at multiple levels. We might ask them. Can you hear me at the back? Yes. I have my teacher voice on. So you start by asking yourself, who am I? And you think we're asking you about your name and what state you're from, what city you're from, but in the biology class this year, we're actually getting a little bit more into it by asking you by this question and giving you the option to look at your genetic information. Right? So you can dig really deep into that question. So far, you have looked at your family tree, correct? Mm-hmm. And you have also looked at your ancestry composition by asking where your ancestors came from as far as the geographic locations go around the world, correct? And now we are getting into the next level of digging into this question of who am I by going even deeper into your ancestors. So we're going into deeper into the human evolution tree. And for that reason, we thought it would be particularly exciting for the biology students to have someone who has expertise into digging deeper into the story. So we found the best storyteller we could think of for your benefit. And I'm so excited that you took the time to come here. So why did we pick this particular storyteller? Why did we pick Dr. Disotell? Let me tell you, we have launched this project with the hopes that you see your education in an interdisciplinary context. So you're asking as a biologist, but also as a person who's situated in society, as a historian of your own self. And we have a very interdisciplinary speaker. Those of you who had lunch with him today know what his disciplinary backgrounds are, right? And I'm going to be a teacher and say, what do you remember? What are the disciplines that he said he was interested in? Biological anthropology, but others too. What was the other thing he was interested in? So primatology, but also computer science and mathematics, paleontology. And it appears that he has now a new expertise, which is arts and being like a TV star, right? So he is a very interdisciplinary person, wears many different hats. If you watch Daily Show, if you watch Comedy Central and watch John Oliver, you might have seen him on his show talking about science. And if you have seen Skype TV, you've probably seen him in Search of Bigfoot, the Bigfoot Bounty TV show, which he talked about with the students today. And he does that as a science advocate, trying to talk to people who are sort of on the borders of science and pseudoscience. He has won 2 Teachers of the Year awards. He really cares about the students, and they know that. But I can also brag about him as an expert in a few different ways. He has top-notch research credentials. He's currently working on a $250,000 NSF project. But the thing that I was most excited to learn about him is that he has actually sequenced Jane Goodall's hair samples, and not just hers, but her favorite chimpanzee, Frodo's hair samples for an Animal Planet show which was aired already. So if you want to see any of this, just Google him and you'll find him on YouTube. And to get to some regular details that you expect to hear in an introduction, let me tell you that he got his bachelor's from Cornell in 1985. His graduate work was completed at Harvard in 1987, and he has been at NYU since 1992. So here we have Dr. Todd Disotell, who's going to be very thorough. He's going to bombard you with information if you don't know that already. But he's also going to be very thoughtful in giving you measured interpretations of the data. It's going to be thoughtful. And I think you're going to have a good time. So feel free to ask him quick questions. Because this is tailored for students, we're recording it. Next year when we do the same class, we unfortunately can't afford to have Dr. Disotell come back to us. So we're going to record this for next year's students. Feel free to ask him a quick question, but let's not get into a back and forth. Is that fair? Without further ado, we have Dr. Disotell for us.
Well, thank you very much for that wonderful introduction. And Dr. Pai had asked me to just say a few things about myself. And so besides doing real science, if you will, I do believe in the public outreach. You can't see this really because my resume of TV shows is actually getting quite long, over 30 different episodes in a dozen different series, where I try to present the face of science to the very broad public, the hundreds of thousands to the millions of people who watch television instead of the dozens to hundreds who sit through lectures in college classrooms and stuff. Science on TV, though, suffers a lot. So they always present us like this, but if you ever walk into a lab and see a real practicing scientist, he or she's more likely to look like that. And that's basically how I look in my day-to-day science. In fact, when people in my department see me in a lab coat, their first question, what are you filming today? Because otherwise, unless the inspectors are coming, the lab coats are on the hook. So today, though, I want to weave an interesting story of our evolutionary history. And so I need to do a few minutes of sort of quick background and then take you into some of the latest things that we've discovered just in the last 6 years that have actually very significantly upended our view of human evolution, and including some mistakes or errors that we make. And if the scientist doesn't make a mistake, he or she's not asking the right questions. We learn from our mistakes, and those are very, very key things. And so I'm going to give you one of my most famous flubs or mistakes in science. And if I have time, I'll go through some other more recent ones. So the human fossil record now, and I noticed some nice fossils in one of the cabinets in the hallway, is actually chock full of different species. At almost every different phase of our evolutionary history in the last 6 million years, there have been 3 or 4 different species alive on the planet, of human or human-like species alive on the planet at any one time. But we thought we were super special, and that for the last 50,000 years or so, or 40,000 years ago, we were the only ones. We were the survivors of this sort of human evolutionary odyssey. And we're going to see in a few minutes that indeed we are not. So I specialize in molecular genetics, even though I've had paleontological fieldwork experience and anatomy and all of that stuff. But we can actually use genetics to answer several questions about the fossil record. I'm not going to talk about the origins of humans separating from chimps 6 million years ago or so. Instead, I'm going to talk about the sort of the present and the last few 100,000 years. Until 1997, we could use genetics to look at modern humans, ourselves, Homo sapiens, and extrapolate from the variation on the planet today backwards in time until our common ancestor, which we'll see in a moment, about 200,000 years ago. And that was it. But in 1997, we were able to extract the first DNA out of a fossil Neanderthal, and that all of a sudden brought Neanderthal evolutionary history into play, the relationships of Neanderthals to moderns. And we're going to see now, we suspect there are 3 or 4 or 5 other species of near humans that lived just in the last 50,000 years or so. And I'm going to weave that story. So 20 years ago, we basically had Neanderthals. This is the actual original specimen found in the Neander Valley, Germany, for which they are named. And we have the so-called Cro-Magnon people. In the last 20, 25 years, we have discovered hundreds and hundreds of new fossils filling in the human fossil record, as well as making it more complicated to interpret. But one of the ways that one of the big advances is we have now realized that anatomically modern people indistinguishable from you and me, go back in the fossil record to 200,000 years ago. When I was an undergraduate, that date was 30,000 years ago with the Cro-Magnon people found in southern France. So the fossil record has revolutionized our interpretations of early human evolution. And we have moved the view that anatomically modern humans first evolved in Europe and spread throughout the world, or spread their genes throughout the world, to realizing that Africa is the home of modern humans, of modernity. The oldest human fossils are about 195,000 years old in Ethiopia. There are 160,000-year-old ones. There are 120,000-year-old ones in South Africa. And out of Africa, the European and East Asian fossils are only 30, 35, 40,000 years old at max. So the fossil record has sort of changed our view of human evolution. And one of the main ways it's changed the way over the last few decades we thought about evolution. From the 1920s until really the 1970s, a view of human evolution known as the multi-regional hypothesis was in the ascendancy. And that assumed that all of the different major groups of people, those from sub-Saharan Africa, those from Europe, those from Asia, those from Australia, actually evolved independently from each other, meaning we were very deeply different from each other. There are problems with that kind of just evolutionary theory, and that's not how species actually evolve, but humans are special, everybody said, and so maybe humans actually did this. But other paleontologists said, well, the way species evolve is you have one subpopulation, begins to diverge, and it either replaces all of the existing ones or it lives side by side. So the opposite of the multi-regional model of evolution was the recent replacement model, that one place evolved into us and then spread throughout the world, replacing all of the other pre-human forms that were out there. And so we countered the multi-regional theory with the recent African replacement theory. But it wasn't until 1987 that the first really significant genetic study came out, and it examined mitochondrial DNA. Mitochondrial DNA is special because it's only inherited in the maternal line. So everybody in here has their mothers and her mothers and her mother's mitochondrial DNA, and it's very rapidly evolving, and there are many, many copies of it in every cell. So in the early days of DNA technology, mitochondrial DNA was the easiest thing to work with. A young graduate student at the University of California at Berkeley named Becky Kahn actually did an amazing study that just blew open genetics for the studies of human evolution, in which she typed the mitochondrial DNA of 143 people representing Africans, Asians, Australians, New Guineans, and people from Europe. And she inferred an evolutionary tree from that, and I hear you're going to be learning how to do that shortly. And she noticed that all of the deepest roots of that evolutionary tree were found amongst people of African descent. That itself isn't surprising. From 6 million years ago to 50 million years ago, or till maybe 1.8 million years ago when Homo erectus left Africa, all of human evolution took place in Africa. So this could have been Lucy, Australopithecus afarensis, or it could have been the first member of the genus Homo, Homo habilis. But it turns out, knowing the mutation rate of mitochondrial DNA, this ancestress, if you will, was only 200,000 years old. And that would mean all of those Homo erectus populations and their descendants everywhere outside of Africa did not leave any mitochondrial DNA behind. And in a poor sense of science reporting, which is almost always the case, the press picked up on a single female ancestor in Africa and dubbed her Mitochondrial Eve. Even made the cover of Newsweek. But it turns out having a single ancestor for any segment of your genome is exactly how it happens. You can trace any gene in your genome and all the variants you find amongst all the people ultimately traces back to a single common ancestor. It could be a bacteria, it could be a 20 million year old ape, it could be 4 million year old Australopithecine, but in this case it turned out to be a 200,000 year old woman in Africa. But she wasn't the only woman. She had sisters and cousins and aunts and others around. It's just her type is the one that survives in us today. And so this was a really groundbreaking study for 1987. Over the next decade, more and more mitochondrial DNA was sequenced, and a pattern kept reemerging that people of African descent were about twice as variable, this is mean percent sequence difference, as people of Eurasian descent. So all over, everybody in Europe and Asia had half the amount of genetic variation found in Africa. More and more studies just continuously confirmed this. In fact, if we divided the world up by our mitochondrial haplotypes, there are 3 major haplotypes. They're all found in Africa. Some of these are subtypes. And everybody outside of Africa, East Asia, South Asia, and Europe, is a descendant of 1/3 of the diversity found in Africa. And that dates to only about 50 to 60,000 years ago. So with new studies where we've sequenced now thousands of people of African descent and all over the world, we can now see the origin of our genus is just under 200,000 years ago, but basically the out of Africa exodus was probably between 50 and 60,000 years ago. So we spent 140,000 years evolving into really diverse people within Africa, and a small subset of that very recently escaped outside of Africa. So if we look at the gene pool, we see a lot more variation in Africa than we see outside. And again, the outside of Africa variation is small. But that's our maternal history. And we all have fathers as well. And so your mother might be from one country, but father from another. So what are you? Well, you're actually a mixture of both. So if we really want to see our history, we need to look at both sides of our heritage. And so if you just look at one, you're going to have a skewed view. If you look at both, you're going to have a more complete view. So if we can look at the maternal side, how do we look at the paternal side? Well, at least amongst males, we can look at the Y chromosome. And the Y chromosome for decades was thought to have almost no variation in it. But as sequencing technologies improved, we began to find a considerable amount of variation in the Y chromosome. And when Y chromosome trees, somebody read that. When Y chromosome trees were finally worked out, Adam was an African. And if we look at the evolutionary history, the Y chromosomes of everybody on the planet appeared less than 200,000 years ago, 50,000 years ago or so. They spread to the rest of the planet. So we have a very consistent story, both of our paternal and maternal lines. Now, we can look at the rest of the genome itself and we see a similar story. But we can actually do something cool, something that Darwin himself recognized. Our parasites co-evolve with us. And so if we look at bacteria found in our stomachs that are passed from mother to offspring, and we reconstruct those bacteria's history, we get an out-of-Africa history. In your mouth, you are teeming with Streptococcus mutans. It's actually the bacteria that cause cavities. If we do an evolutionary history of Streptococcus mutans, we see deep African roots and shallow Eurasian roots. So any segment of the genome, or most segments of the genome that we look at, we see this very consistent phase of the origin of modern people from about 200,000 years ago to the first migration out of Africa less than 100,000 years ago to ultimately migrations into the Americas maybe only 15 or 20,000 years ago for people of Native American ancestry. And just genetic system after genetic system ruled out this multi-regional model of evolution. And the question was this out of Africa exodus a complete and total replacement of all of the descendants of Homo erectus elsewhere in the world, or when they ran into the Neanderthals in Europe and other populations, did they interbreed a little? And until, again, about 2006, or actually, no, I'm sorry, 2010, that's how things stood. So I did some various analyses and wrote up, here's the story as we know it. But the big question still remained. These guys, Neanderthals, this is a Neanderthal from Gibraltar in Spain. This is, again, the German Feldhofer 1 specimen from the Neander Valley. This is a French Neanderthal called the Old Man. He basically wasn't actually, he was only in his 40s, but he had arthritis and life was tough back, 100,000 years ago. But something interesting had also been noticed. Neanderthals and modern humans actually overlapped in time and space for a considerable amount of time. In some regions, maybe 5,000 years, but that's a long time. And in other regions, maybe up to 50,000 years. And the question was, if these two types of people lived together or overlapped, did they ever mate and produce offspring? So this question was attacked. People looked at the fossils and tried to interpret, this looks like a hybrid. That's really difficult to do morphologically. But in 1997, Svante Pavo and colleagues were able to get permission to take this gigantic chunk of bone out of the original Neander Valley specimens, right humerus, and then using PCR, or the polymerase chain reaction, they were able to actually amplify a small segment of the mitochondrial DNA. A lot of that DNA was contaminated, but they had a consistent signal of a new sequence that basically did not overlap with modern humans. And when they inferred an evolutionary tree from that, this Neanderthal fell outside the range of all modern variation. The theory of the people who believed in hybridization was, well, if Neanderthals were only known in Europe, then Europeans should have some Neanderthal DNA. They don't have any mitochondrial Neanderthal DNA at all. people dismiss this like, oh, it's a single sample, it's one individual. More and more studies, this slide is actually from the second study. This is a slide, Neanderthal from Mesmeskaya Cave in the Caucasus Mountains of southern Russia. Shortly thereafter, 2 more Neanderthals were sequenced, and the Neanderthals all clustered together to the exclusion, not just of Europeans, but of all modern peoples. And so a growing consensus began to arrive that Neanderthals were significantly different genetically from modern humans and were unlikely to mate. Part of the problem is ancient DNA is almost always contaminated with modern DNA. The person who excavated it, the person who studied it in the museum, somebody in the lab analyzing it. And so we never thought we would be able to get early